Hello, everyone, and welcome to the Life Raft Group's virtual Life Fest weekend. My name is Diana Nieves, and I am the Senior Director of Outreach and Engagement here at the LRG. Thank you to our sponsors, Decipher, Genentech, Novartis, and Pfizer for making this weekend possible for our community. Please visit our website for more information about the weekend, about our Just Do It Walk, and Just Awareness Day. And don't forget to check out our store to order masks, t-shirts, cookbook, and just art. Today, I have the honor of introducing Dr. Josephy. David is a professor of biochemistry at the University of Guelph in Canada and serves as president of the Life Raft Group Canada. David will be presenting on GIST 101, the biology of GIST today. Welcome, David.
Thank you, Diana. So today I'm going to give a version of a talk I've given a few times, but it's been updated a little bit with more recent information. So I'm going to talk about the biology of GIST. Along the way, I'll say a little bit about the medicine related to that biology. Let me say at the outset, as a disclaimer, that I'm not a medical doctor, I'm not a physician, and nothing in this presentation should be taken as medical advice or as a substitute for consulting with your doctors. Here are a few of the topics I want to talk about today. I'll say a little bit about what causes GIST. Then I'll discuss in some detail where GISTs come from in terms of the cell biology. Then we'll turn to talking about KIT, the protein and gene that is of great importance in GIST, understanding GIST. Along the way, we'll have a short crash course in molecular biology to get you up to speed on some of the terminology. Finally, I want to turn to the issue of drug therapy, very important question in GIST treatment. We'll talk about TKIs, I'll explain what they are and discuss a little bit about how they work. And finally, I want to tell you a little bit about the new generation of GIST drugs. Let's begin then by discussing what causes GIST. Well, the simple answer is we generally don't know. The great majority of GISTs, not quite 100%, but almost, occur sporadically, that is randomly. They're not inherited and they can't be passed on. As far as we know, GIST strikes completely at random. There are no known environmental, geographical, occupational, dietary, lifestyle, or other causes of sporadic GIST. And as I usually say at these Life Raft group events, if there were any obvious factors in common, any obvious risk factors, we would have noticed them by now. So GIST really does strike at random. The small exception to that is that there are a few cases of GIST, just a few percent, which are clearly related to inherited predispositions, genetic predispositions. And these are associated with a few particular genes, like the NF1 gene, which is related to a condition called neurofibromatosis, and also SDH deficient GIST, and the very rare cases, just a few families known around the world, where KIT mutations are inherited. These are discussed in a paper that is referenced on this slide. I'm not going to say anything more about these syndromic forms of GIST, these familial forms. They will be discussed in other events at LifeFest, but this presentation today is limited entirely to the prevalent major form of GIST, which is the sporadic GIST that is not inherited. Okay, let's talk about where GISTs come from in terms of the cell biology of GIST. First, recognize that there are many hundreds, maybe thousands of different types of cells in the human body, and they look very different, and they have very different functions. And I've just shown you a gallery of a few of these to indicate just how different human cells can be. muscle cells in the heart, hepatocytes, the functional cells in the liver, adipocytes in fat tissue, and all the different, some of the many different forms of white blood cells. These cells look differently, they behave differently. Almost any cell in the body can, in principle, give rise to a cancer. And though the cancers that arise from these very different cells are just as different biologically as the cells themselves. Cancers can begin in almost any type of cell in the human body. It is fundamentally the type of cell from which cancer develops that defines the cancer, the biology of the cancer, and therefore that determines its course of treatment. Pathologists are the doctors who are tasked with the job of identifying the cell type and therefore identifying a type of cancer. And they usually do that by studying a specimen taken at a biopsy or at surgery. I'll say a little bit later about how that's done. And then that information is provided to the surgeon and the medical, or most importantly, to the medical oncologist, who will use that information to plan the course of treatment of the cancer. Let me emphasize again, because it's often a cause of confusion. It's the cell type, not the organ, that defines a cancer. Let's give a couple of examples unrelated to GIST. Think about skin cancer. It's the most common form of cancer. People talk about skin cancer a lot. We're all aware of the danger of sun exposure and so on. But there are multiple different kinds of skin cancer. The common and relatively easily treatable skin cancer is a basal cell carcinoma. But there's another kind of skin cancer that you've probably heard of, malignant melanoma. That's another skin cancer, but it's a totally different disease, different treatment, different prognosis, different biology. If you look at the lung, we talk about lung cancer. It's a common cancer, especially in cigarette smokers. But the common cancer in cigarette smokers is a lung adenocarcinoma. You probably heard about mesothelioma, which is associated with asbestos exposure. Again, that begins in the lung, but the carcinoma and the mesothelioma are completely different diseases. They're treated differently, different prognosis, and so on. So I hope that point is clear. It's the cell type that matters. In fact, all cancers are categorized according to the histological type of cell in which they arise. And there are two major kinds we need to think about here, carcinomas and sarcomas. These are two different general classes of cancers. Most of the common cancers you will hear about in the press or that you read about in the media or see it discussed in TV and so on. Most of these common cancers are what we call carcinomas. These arise from cells that are in the lining, the outside lining of the human body, including the internal linings of the gut, for example. So this includes the most common cancers that one hears about: skin cancer, most forms of lung cancer, most forms of colon, breast cancer, and so on. These are more than 90% of human cancers are carcinomas. GIST is not a carcinoma. It belongs to a different category of cancers called sarcomas. Sarcomas are cancers that arise not from these lining tissues, but rather from the deeper connective tissues like muscle, cartilage, and bone. Sarcomas in humans are rare. They're only about 1% of human cancers. Turns out they're more common, for example, in our friends, the dogs. Canine sarcomas are quite common, but in humans, sarcomas are rare, maybe 1% of human cancers. That's why treating sarcomas is really a specialty among oncologists. Most oncologists rarely, if at all, treat sarcomas. And within sarcomas, treating GIST is even a subspecialty. That's why we advise people to see a specialist oncologist who has seen a lot of GISTs in their career. Okay, GIST is a cancer of the GI tract. Let's talk a little bit about the gastrointestinal tract as shown in this picture. So it is long, continuous tube that runs from the mouth to the anus, and that includes the digestive organs, the stomach and the intestine. In fact, carcinomas of the gastrointestinal tract are relatively common. Stomach cancer and certainly colon cancer is one of the most prevalent cancers, but these are carcinomas. GIST is a GI tract cancer, but it's a sarcoma, not a carcinoma. So it's much rarer than stomach cancers, colon cancers, and so on. Suppose we were to make a cross-sectional cut of the gastrointestinal tract. That's shown in two different views here, the cross-section on the left and a sideways view on the right. We're talking about one long continuous tube. The center of that tube is what we call the lumen. It's the open space through which the food and digestive material is passing. Surrounding it is a layer of epithelial cells, and surrounding that are supportive tissues like muscle. And that's true all the way along the GI tract. Topologically, the inside of the GI tract is really the outside of the body because the food passing through your gut, it's not part of your body, it's really external to the body. Even though it's within the interior of the body, it's topologically outside. And that's why the innermost layer of the GI tract is epithelial tissue. It's almost like skin tissue, but on the inside of the GI tract. Here's another view, another cross-sectional view in a little more detail. Now, the point I want to make here is that carcinomas, the more common GI cancers like colon cancer, they begin in the epithelial lining, the outside surface of the body, which is to say the core, the inside, next to the lumen of the GI tract. That's not where a GIST begins. GISTs are sarcomas, and they begin, as shown by the red star here, they begin in the muscular wall that surrounds the GI tract, the supportive muscular tissue around the core of the GI tract. So that's a very important difference between the sarcoma and the more common carcinoma. And this has a lot of implications for surgery and treatment of GIST. So why do they start in the muscular wall? Well, let's talk a little bit about the function of the GI tract. The GI tract mediates a process called peristalsis. Peristalsis is the process that moves food through the GI tract, through the digestive system. It's coordinated waves of muscle action that squeeze the GI tract, that squeeze the tube, and push the food through the tube. And that's done by the muscle surrounding the GI tract, surrounding the intestine, for example, rhythmically squeezing, contracting to push the food through or the digestive material through. These are waves of contraction called peristalsis. Why does this matter? Well, for these waves to occur in a coordinated fashion, you need a pacemaker. You need a cell that is calling like the coxswain in a rowing team is calling stroke every few seconds to tell people to put their oars into the water. You need a team coordinator, a coach, that is telling the muscle when to contract in these rhythmical waves. And that coach, if you will, is a very specialized kind of cell. It was first identified by a famous Spanish histologist many years ago named Cajal. And these cells are called the ICCs, the interstitial cells of Cajal. These are the cells where GISTs begin. They are the pacemaker neuromuscular cells that coordinate the peristalsis throughout the gastrointestinal tract. And there's a schematic view here that shows you the way that these ICCs surround the GI tract. These cells are neuromuscular. They send out electrical signals. In fact, we can record those electrical signals as a matter of interest. This shows you the rhythmical pulsing or electrical signals being sent out by the ICCs, in this case in a mouse. Now, an important consequence of this peristaltic wave runs right through the GI tract from the beginning to the end, from the esophagus to the rectum. So these ICC cells are found right along the entire GI tract to coordinate the contractive waves. Because the cell type is found right along the GI tract from one end to the other, that means that in principle, a gist can arise anywhere in the GI tract. And in fact, they do. As you can see in this diagram again, I'm indicating on the right in the boxes, the approximate percentage of gists that are found beginning in particular organs, primarily the stomach, but also in the small intestine, the large intestine, the rectum, and even the esophagus. So important to recognize that wherever the gist begins along the GI tract, it's still a gist, regardless of where it's located along the GI tract. There are some differences in the behavior and treatment of gists in different organs, but fundamentally it's the same kind of cancer. Say that one more time to emphasize the point. A gist that starts in the stomach, it's a gist. It's not what people usually refer to as stomach cancer, which is a more common adenocarcinoma. But when we see stories about the Life Raft group or about GIST in the popular press, the journalists always call it a stomach cancer, but it really shouldn't be called a stomach cancer, even if it begins in the stomach. It's a very distinctive kind of cancer, the GIST sarcoma. Similarly, if it begins in the colon, let's not call it a colon cancer. That's a common colorectal carcinoma. GIST is GIST. It's a very different disease. GISTs, like other cancers, can metastasize, that is, they can spread. Cells can be shed from the primary tumor and begin to grow in new sites in the body. GISTs in particular tend to spread into the liver and into the peritoneum, which is the membrane lining the cavity of the abdomen. Now, a very important distinction that most of you are probably aware of, but it's worth reiterating. When a GIST is diagnosed, when a doctor finally recognizes the condition as being a GIST, that may occur at a time when the GIST is still localized, just growing in one spot, or it may not be diagnosed until the GIST has already become metastatic and spread. If the GIST is localized, then in principle and often in practice, it can be completely cured by surgery. The surgeon will remove the entire tumor and it will never recur. That's possible. However, in many cases, even after the tumor has apparently been completely removed, you still have a recurrence, presumably because a few cells had already been shed by the time of the surgery, and the surgeon can't remove every cell if it's spread throughout the body. If the GIST is already recognizably metastasized before, at the time of diagnosis, if metastases are already evident at the time of diagnosis, then certainly that GIST cannot be cured by surgery because it is spread. Surgery might still be performed, but systemic therapy will be required, and that is usually going to be drug therapy. So this distinction between localized and metastatic GIST in terms of treatment of the disease is a very important distinction. Well, here's an old record by Sting, his song Englishman in New York. And I put it up there to remind us that if an Englishman goes to New York, he's still an Englishman. Why do I put that up? Well, to indicate that metastases, wherever they travel, wherever they occur, they still have the biological properties of the primary tumor. Very important. This is a common misconception here. People fail to recognize this. So GIST metastases in the liver, for example, or in the lung, they're still GISTs. They have to be treated as GISTs. They're not liver cancers or lung cancers. And unfortunately, people often get confused, and we hear people say, well, I was diagnosed with GIST 5 years ago, but now I've got liver cancer. But what they normally mean, actually, is that they now have metastases of the GIST in the liver. And those metastases have to be treated as GISTs. They're not liver cancer. Liver cancer would be a cancer arising from the cells of the liver. So it just, and wherever it goes in the body is still a GIST. An Englishman in New York, still an Englishman. Okay, let's change gears now and start moving towards some molecular analysis of GIST. Let's talk about the KIT protein and gene. And then we'll have to talk a little bit about the basics of molecular biology, a little bit of a crash course, so we can talk about the molecular biology of GIST. Everything changed in 1998. Any research or medicine about GIST before 1998 is basically obsolete now because of the breakthrough that was made in that year, but particularly by these Japanese doctors to whom we owe a great debt of gratitude, these doctors in Osaka, Japan, who discovered the most fundamental property about GIST, which had not been known beforehand, that GIST cells express a particular protein called KIT, which in fact very few other cells in the body do, and that in most cases of GIST, the gene encoding KIT has undergone a mutation, producing an aberrant form of the KIT protein, and that mutation drives the formation and growth of the GIST cancer. All of that discovery goes back to 1998, and it's really revolutionized our understanding of GIST and made a huge advance with regard to GIST treatment. So we owe, as I say, a great debt of gratitude to these scientists and doctors. Well, that was 1998 and the breakthrough, but things have moved a long way since then. Here's an update from the point of view of 2020. What appeared to be a single uniform disease called GIST, we now recognize that it's really a bit of an umbrella term. In fact, there are quite a few different forms of GIST, maybe more than a dozen different forms. They're all closely related. They all seem to begin in pretty much the same cell type, but at the molecular level, they're distinct. So I said that in 1998, it was discovered that most GISTs have a mutation in the KIT gene, as I will explain in more detail in a few minutes. But in fact, we now know that that's only about 3/4 of GISTs. About a quarter of GISTs do not have a mutation in that gene. And we're gradually figuring out what's going on with those other forms of GIST. For example, about 15% have a mutation in a gene related to KIT called PDGFRA or alpha. And there's another 10% or so that we used to call wild type GISTs. These are quite distinct diseases in terms of their biology and treatment. And I'm going to say little or nothing about them in the remainder of my talk, but you will hear more about those conditions in other talks at LifeFest. Okay, well, I earn my salary by teaching biochemistry and molecular biology, so let's do a little bit of that. Let's go back to the beginning of molecular biology and think about genes and proteins. Genes encoded in DNA are the codes, if you like, the blueprints for the construction of the proteins that make up the cells of the human body. There are more than 30,000 different kinds of proteins encoded on the human genome. We saw earlier at the beginning of the talk, I stressed that different cells look very different. A blood cell or a muscle cell or a fat cell, they look very different. What makes them different fundamentally is that different cells make or express, to use the technical term, different cells express different proteins, different sets of proteins. So by analogy, we can say that the human genome is the library of all possible proteins, but different cells prefer to read different books. They make different choices out of the library. For example, a muscle cell makes muscle proteins like actin and myosin. Red blood cells make hemoglobin and some other proteins too. So different cell types make different kinds of proteins. Looking at that another way, genes and proteins. So genes are made out of DNA, the sequence of the 4 bases, A, C, G, and T, as shown at the top of this picture. Those genes are used to make an intermediary molecule called RNA, ribonucleic acid. And in turn, RNA is used as the blueprint to construct proteins, as represented by the squiggle diagram at the bottom of the picture. And proteins are made out of amino acids. Proteins are linear chains of amino acids. There are 20 different kinds of amino acids. Some of you may have learned this in a college course. The 20 amino acids have historical names, really, but they each have specific names: alanine, cysteine, glutamic acid, tryptophan, and so on. And since there are 20 different amino acids, and there are 26 English letters, we can assign a distinct letter to each amino acid, and that's done in the so-called one-letter code. Often it's the first letter in the name of the amino acid, but not always, because there are some ambiguities. So a protein then is a sequence of amino acids, or amino acid residues, as we say. And that sequence could be perhaps 100 amino acids long, or anywhere up to thousands or even tens of thousands of amino acids in a specific linear chain. This is the protein that is of most interest when we're discussing KIT. This is a sequence of the KIT protein. It happens to have 976 amino acid residues in the protein, and they're shown here, beginning at 1 with the methionine, M, and ending with the valine at number 976. And the normal wild type functional KIT protein has exactly this sequence and no other. Now, a little bit about protein structure, because we need to think about this if we want to think about exons. And exons are a very important theme in understanding GIST. So proteins can be very long chains of amino acids, almost 1,000 in the case of KIT, in some cases several thousand. But we can often think of proteins as being made up of several connected sections, each of which is a little bit different, and we call those subsections or substructures, we call them domains of the protein, typically say 20 to 30 to 100 amino acids in length. So I've indicated this by some colored bars, and just as a metaphor, you can think of the southern US as being, if we were meeting in New Orleans, unfortunately we're meeting virtually, but if we were in New Orleans, you'd recognize that we have a series of states: Texas, Louisiana, Mississippi, Alabama, Georgia, each of which is distinct, but which are linked together in a particular sequence, forming the southern tier of the USA. In the same way then, each domain of protein structure is a separate segment of the gene coding for that protein. And these segments of the gene, corresponding to the states, if you will, we call them exons, for reasons I won't go into here. The KIT gene is made up of 21 different exons. So to continue the analogy, you can think of the human genome as being the library. I've said that particular cells choose to read particular books, that is, to make particular proteins. Within that book, you can think of an exon of the gene or the corresponding domain of the protein as being a particular chapter in that book. The KIT book has 21 chapters. And then an amino acid is a particular letter within that chapter. This is the KIT protein. Again, the sequence again. But now I've indicated the 21 exons. I've highlighted every second exon so you can see where they begin and end. So the sequence is really subdivided into 21 different sections. And here they are numbered from 1 to 21. So why am I belaboring this arcane molecular biology? Well, the reason is that each of these exons corresponds approximately to a particular region or a functional piece of the overall KIT protein. And so here I'm showing you a picture from a manuscript that shows you a schematic view, a cartoon view of the KIT protein. It's a very large protein. It sticks right through the membrane of the cell. That's indicated by the blue balls in the middle. The membrane is indicated there. So it has a number of domains, a region of the protein that protrudes outside the cell, the extracellular domain. It's got a part of the protein that goes through the membrane, and then it's got a part of the protein inside the cell. And these particular regions of the protein are encoded by particular exons of the gene. So many of you will have heard people talking about exon 9 mutations, for example, exon 11 mutations. We'll get into that in just a few minutes. So exons of the gene correspond to particular domains or regions, functional regions of the KIT protein. So a little bit more about KIT. Sometimes you will hear it called c-KIT, which just means cellular KIT. And sometimes it's referred to by an inscrutable telephone code, area code, CD117, but that's really just another way of saying KIT. So the KIT protein is made by only a few types of adult cells, including the ICCs, the cells of Gehal, and including almost all GISTs. The pathologist is tasked with identifying a cell or a tumor as being a particular type of tumor, for example, identifying a GIST. And the key tool in doing that is a technique called immunohistochemistry, or IHC. And I won't go into details about that, other than to say that this is a technique for determining whether a particular cell, such as a particular tumor sample, expresses particular proteins. And if it's a GIST, it almost certainly expresses KIT, and if it's not a GIST, it almost certainly doesn't. That's true whether or not the KIT is mutated. Whether it's the wild type or the mutant form of KIT, it's normally going to be expressed by, usually going to be expressed by any GIST tumor cell. So to do this analysis, the pathologist makes a thin slice of the tissue sample obtained from the biopsy or the surgery, and then stains it with an antibody that recognizes the KIT protein. And it's then treated with a series of chemicals such that when the antibody binds, it leads to the formation of a brown stain that you can actually see, the pathologist can see under the microscope. And here's an example of that, a photomicrograph shown at the bottom. And all of that brown staining that you can see in this image represents KIT protein expressed by the GIST tumor cells. Very important to make sure that this is done in the pathology of a GIST. It certainly will be done. It's a standard procedure. And if you see your pathology report, it will almost certainly say that the tumor is KIT positive. That is, it does express the KIT protein. That tells you that it's a GIST. It doesn't tell you anything more than that, but it tells you that the tumor is a GIST. Okay. KIT is a protein belonging to a very large family of related proteins, like a big clan. And these proteins belong to a class of proteins which we call tyrosine kinases, TKs. I'm not going to describe all of that in detail, but let's accept that they're related, a lot of related proteins called tyrosine kinases that perform very important functions in cells. So what I'm showing you here is simply a family tree. It's a genealogical tree, if you will, of the relatedness of many of these different human tyrosine kinase genes and proteins. And I've indicated KIT in blue up at the top right as being one of these many tyrosine kinases. You can see that there's a closely related family, the PDGFR family, and I've mentioned PDGFR alpha earlier. You see that's quite a closely related tyrosine kinase. Much more distantly, in a much more distant branch of this tree, is another protein called ABL, and that will be important for reasons we'll see in a moment. It's another tyrosine kinase, but quite distantly related to KIT. The KIT gene in a GIST is usually mutated. So what is a mutation? A mutation is simply a change in the DNA sequence that encodes a particular protein. Mutations occur randomly, but their effects are not random. Certain mutations will lead to a cell carrying that mutation to die. Other mutations may cause a cell to grow and divide faster. Let's talk about KIT mutations. I've said now the KIT protein is a particular kind of protein. It's an enzyme, a protein catalyst of a family called tyrosine kinases. And these enzymes, these proteins, act on other proteins. They trigger what we call a signal transduction cascade. That's a topic that we won't go into in detail in this talk, but a signal transduction cascade is a series of molecular events which motivates, if you will, a protein to undergo certain changes, functional changes, like causing cell division, for example. In about 3/4, about 75% of GIST cases, not 100%, but in most GISTs, the KIT gene is mutated. That mutation leads the cell to make an aberrant or different form of the KIT protein. And the KIT gene is what we call an oncogene. That's a gene which, when it is mutated, encodes a protein product that instructs the cell to keep on dividing. The good metaphor for that is that it's like a stuck gas pedal. The KIT gene, the KIT protein normally acts like a gas pedal that the cell can step on to divide, and it can then release the gas pedal to stop dividing. But when the KIT gene has been mutated, it acts like a gas pedal that's stuck to the floor. And it keeps on fueling the engine, whether it should be or not. And so we say that the mutated KIT gene, the resulting KIT protein, is a driver that tells the GIST cells to keep on dividing and proliferating. That's the fundamental discovery that was made in 1998 that transformed our understanding of GIST. Let me emphasize that the KIT mutations occurring in GIST are almost always somatic mutations. They're mutations that occurred in the tumor. They didn't occur in the germline. So taking a blood sample, for example, will not reveal the KIT mutation. You have to get a sample of the tumor itself. These mutations do not affect the germ cells. Consequently, then, to allay the fears that some patients will have when they're diagnosed, the somatic mutation has not been and cannot be passed on to a patient's children. It's only found in the tumor cells. Now, studies since 1998, increasingly sophisticated molecular studies, have shown us that there are many different mutations occurring in GISTs. Most of those are KIT mutations, but as I mentioned earlier, we sometimes see driver mutations in other genes, like BTGFRA. And the site of the mutation is very important to determine because it affects the prognosis and it affects treatment and the response to drugs. So we strongly feel at the Raft group that mutational testing of the tumor DNA, should be performed on all new GIST cases. Bear in mind that requires having access to a sample of the tumor, usually obtained at surgery. It can't be done simply on a blood sample because the mutations are only found in the GIST itself. In fact, the GIST patient support groups around the world made a declaration of the importance of mutational testing more than 10 years ago at a meeting in Baveno, Italy. So pathologists have now examined in great detail the mutations found in KIT and related genes in GIST. And as many of you will know, the most common mutation shown here is the exon 11 mutation. That's more than half of the mutations occurring in GIST. Another common mutation, less common but still quite common, is the exon 9 mutation. And then there are other rare mutations like in exon 13 and 17 and PDGFRA. Well, I talked about exons a little bit earlier on. Now, why would it be the case that even though there are 21 different exons in the KIT gene, more than half of the mutations that drive GIST are found in exon 11, and most of the remainder are found in exon 9? Why this preference for those particular genes? Well, exon 9, highlighted here, and the nearby exon 11, these are the regions of the gene corresponding to the regions of the protein where most of the primary mutations in GIST are found, and there's a reason for that. Right? So here are the regions of the protein sequence, and here are cartoons of the protein structure. Cartoon we showed earlier on the left, and a different representation shown on the right. The KIT protein spans the cell membrane from the inside to the outside. There's a little portion of the protein, it's actually exon 10, that crosses the membrane. Exon 9 and exon 11 are the regions of the protein just inside and just outside the cell membrane, and these turn out to be functionally critical for the protein. In particular, exon 11, the so-called juxtamembrane or next to the membrane domain of the KIT protein, it's really part of the switch that switches KIT in between two different shapes or conformations, as we say, a form of KIT that is inactive biochemically and a form of KIT that is active and which signals the cell to grow and divide. And so mutations occurring in particular portions of exon 11 tend to flip this switch and flip the KIT protein into its, as we say, constitutively active, always turned on, stuck gas pedal form. And so that's why mutations in exon 11 are so important. If you have had mutational testing done and you look at your pathology report, you may see an indication of the mutational testing results saying what the mutation is in your particular GIST. For example, you might see KIT V560D. That's what we call a missense mutation. It's a terminology that indicates that the amino acid 560 out of the nearly 1,000 residues in amino acids in KIT, building block 560 has been changed. The normal building block is a V, that is a valine, and it's changed to a different residue. It happens to be aspartic acid, represented by the symbol D. Another common mutation you might see is KIT W557K558 del for deletion. So this is a deletion mutation. The 557th and 8th amino acids are simply absent, and that's indicated schematically here. So here at the top, I've done a color-coded little section of the KIT protein. Each colored block representing a particular amino acid in the sequence. The wild type sequence, the normal sequence, is shown at the top. E, V, Q, W, K, V, V, E, E in this part of the protein. So a missense mutation, as we just looked at, would look like the middle picture. The V, the third V in the sequence, has changed to a D. The deletion mutation would correspond to the removal of the W and the K building blocks, giving a slightly shorter protein, missing those building blocks. So you'll see that kind of terminology referred to with regard to KIT mutations and other mutations in GIST. Okay, in the last section of the talk, then, I want to talk about the drugs, so-called TKI drugs like Gleevec, and then finally we'll talk about the new generation of GIST drugs. So building on the discovery of the role of KIT in most GISTs, this led rapidly to the introduction of targeted molecular therapies, drugs for treating GIST. This is an image of a PET scan of of the first GIST patient treated with imatinib back around 2000. Before and after. On the left, you see a whole lot of hot spots in the abdomen, which represent actively growing GIST metastases. And after drug treatment on the right, all those metastases have gone cold and stopped growing. It was a remarkable response, the beginning of the imatinib revolution in the treatment of GIST. So let's talk briefly about where these drugs came from, these so-called targeted drugs. It's a remarkable story. It begins with a story that has nothing to do with GIST. It began with a whole lot of research going on over many years on an unusual blood cancer, a leukemia called CML, chronic myelogenous leukemia. It's a blood cancer that clinically is completely unrelated to GIST, but it turns out that the two diseases actually have a similarity or relationship at the molecular level. I already alluded to that earlier in the talk when I showed you the family tree of the tyrosine kinases. So the mutation that causes CML is in a gene called ABL, and ABL is a distant cousin, but a cousin nonetheless, of the KIT protein. Like KIT, ABL is a tyrosine kinase enzyme. It's another member of that family. Drugs that can turn off the activity of those proteins, those enzymes, inhibit those enzymes, are therefore called tyrosine kinase inhibitors, or TKIs. Well, this is a picture of one of the heroes of this story, Dr. Brian Drucker at OHSU in Oregon. He was really the pioneer of the development of targeted therapies for chronic myelogenous leukemia. Really, he ought to be getting a Nobel Prize one of these days, but until he gets that, he'll be satisfied with the Sjöberg Prize, another Swedish prize that he received last year. He got a half share of that, so that's $500,000, and he deserves every penny, because his discoveries with regard to CML, which were revolutionary, have had as one of their after effects the development of imatinib for GIST. So we owe him a great debt of gratitude, even though he never directly worked on GIST himself. So this is the drug that Drucker and his colleagues developed as an inhibitor of the ABL oncogene for CML. It's the drug that's been called the magic cancer bullet. It's completely transformed the treatment of CML. And along the way, it also completely transformed the treatment of most GIST. The molecular structure of the Gleevec molecule is shown in this picture. It's a relatively small molecule. That's why it can be taken as a pill. It's simply a capsule that you can pop into your stomach and digest and absorb out of the GI tract. It's an orally available drug. Okay, so GIST therapy benefited from these CML-related discoveries. Why? Because it was recognized in 1998 after the role of KIT in GIST was recognized. People saw right away that KIT, and for that matter PDGFRA, are, like ABL, tyrosine kinase enzymes. And it turned out that the drugs that were originally developed to inhibit other tyrosine kinases, like the ABL tyrosine kinase, also worked pretty well for GIST. So we now have a whole first generation, as we can call it, of GIST drugs, like imatinib, sunitinib, regorafenib. All of these were developed for other cancers, not for GIST, but they turn out to work reasonably well for GIST. As I'll talk about it later towards the end of the talk, we're now seeing the emergence of a new generation of drugs, what I can call bespoke or tailor-made drugs that were actually designed for the KIT target and the PDGFRA target in GIST. These are just arriving on stream now. So the first three TKIs, tyrosine kinase inhibitors that were approved for use in GIST, these were imatinib, developed by Novartis, brand name Gleevec. Sunitinib, brand name Sutent, developed by Pfizer. And regorafenib, brand name Stivarga, developed by Bayer Pharmaceuticals. These are now the standard first, second, and third lines of treatment for metastatic GIST. By the way, all the names end in ib. This is now kind of a naming convention. The ib comes from the word inhibitor. So these are all enzyme inhibitor drugs, and the standard of naming these drugs with an IB ending has caught on. Okay, all of these drugs act by the same general mechanism. They block the supply of fuel to the protein. The protein relies on a fuel, a molecule called ATP, and all of these drugs act by preventing the binding of ATP. The new generation of drugs, in fact, can use different mechanisms of inhibition at the molecular level. I'll say a little bit about that later. So here's a cartoon. This is another cartoon of the KIT protein. In fact, two molecules of KIT come together to form the functional unit that's shown by the left and right-handed purple cartoons here. So on the left side, then, the left panel is the KIT protein dimer, the two purple units. In the untreated case, in the active GIST, and you see that each of those units binds this molecule ATP and uses it to phosphorylate another protein, and that is the signal that turns on the GIST cell and makes it proliferate and divide. On the right, you see what happens when we treat with imatinib. Imatinib occupies the site on the protein where ATP would normally bind, blocks the binding of ATP, blocks the kinase activity of the KIT protein, and that in turn stops the cell proliferating, and generally it leads the cell to a cell death by a process called apoptosis. So this is our somewhat simplistic but basically accurate view of the mechanism of action of imatinib. Okay, the introduction of imatinib for GIST therapy around the year 2000, 2001 revolutionized, enormously improved the treatment, the therapy of most GIST tumors. However, as many of you already know, the success of imatinib and related drugs has not been enough to cure GIST. We need more drugs, we need more approaches. Why is that? Here are a few reasons. First of all, some GISTs just don't respond to imatinib. They may not even respond to the other drugs. The most common example is a relatively common mutation in the PDGFR alpha gene, the so-called D842V. It really doesn't respond to imatinib, unfortunately. Secondly, we need more drugs because, as with any drug, there are side effects. They can be quite variable from person to person. Some people just don't tolerate imatinib or don't tolerate other drugs. So we need more drugs for that reason. But most importantly, although imatinib slows down or even halts the growth of most GISTs, it doesn't completely eliminate them. Some proliferation probably continues at a slow pace, and over time, GIST tumors have an unfortunate tendency to become resistant to the drug, resistant to imatinib. That's true of a lot of cases of cancers and drugs. And we now have a pretty good understanding of why and how that happens. It happens primarily due to additional mutations arising in the metastases from the original tumor. Okay, we now know quite a lot about the secondary mutations that lead to imatinib resistance. And so here's another cartoon of the KIT protein, the extracellular exon 9 at the top, then the cell membrane, then in fuchsia here at the exon 11 block. I've said earlier on that most of the primary mutations that give rise to GIST occur in exon 9 or in exon 11. But the secondary mutations, the additional mutations that develop and which confer imatinib resistance, they don't occur in exon 9 or exon 11. Typically, they occur in the intracellular exons, especially, as you might guess, in the ATP binding region. So mutations in the region that binds ATP can block the binding of imatinib and reactivate the protein. And so these occur typically in exons 13 and 14, and also in a region called exon 17. So it's excellent that we now have detailed molecular understanding of some of these resistance mechanisms. And one would hope that building on that understanding, we could design new drugs that would target these additionally mutated forms of KIT. And indeed, that's the work that's been going on for quite a few years and that is beginning to come to fruition now. So here's a chart taken from a paper by Jonathan Fletcher and colleagues. It's a sort of traffic light view, although for some reason they use red and blue instead of red and green. But it's a kind of traffic light view of the different mutations, primary and secondary mutations occurring in KIT, and how they affect sensitivity to imatinib. So these are the various secondary mutations shown down the left-hand column from V654 to A829 amino acid residues. And then each column represents a different drug. Imatinib, sunitinib, regorafenib, these are the first, second, and third line drugs. And also shown here are a lot of other TKI inhibitor drugs, which aren't specifically the first, second, or third line treatments of GIST, but they're really been developed for other cancers, but some of these drugs have been used, as it were, off-label for treating GIST. Okay, the first column labeled imatinib. Every traffic light is red. That's because, by definition, this is an analysis of the imatinib-resistant mutations. So, of course, all of these mutations are imatinib-resistant. A red traffic light, imatinib is no longer an effective therapy when any of these secondary mutations have occurred. But you'll see as you move along to the second and third column that some of these mutations are still sensitive to sunitinib or regorafenib, and to some extent the other drugs as well. So in principle, if one of these secondary mutations is developed, we can turn to the second or third line drugs. Unfortunately, with ongoing treatment with more and more drugs, one tends to get more and more different mutations occurring, and gradually the tumor can become resistant to all of these therapies, all of these drugs. Last topic I want to talk about today, and I'm sorry I've gone on a little bit more than an hour, but I'll try to wind it up now. But this is an exciting topic. I want to tell you about the new generation, the tailor-made or bespoke drugs really designed for therapy of GIST in particular. So we're now having a second revolution, if you will, a second revolution in treatment options for GIST. This is just a quotation from a paper that just came out this year talking about the latest developments in GIST therapy. It says the treatment of advanced GIST is rapidly evolving with the development of new novel molecular compounds such as avapritinib and ripretinib, which I will talk about in a moment. The availability now of more than 5 lines of treatment, the original 3 plus these 2 new ones, is likely to shift the second and third line treatment options. For GIST patients with the D842V mutation, which did not respond to imatinib, now avapritinib is available and will probably become the first line therapy for that molecular subgroup. Second and third line treatment, we're still waiting for further clinical trials, but it looks like these too can be tailored to particular secondary mutations found in imatinib-resistant GIST. So let me just say a few words in closing about these various new drugs. Ripretinib, previously called DCC 2618, now has the brand name Quinlock. It was developed by a company called Deciphera. This was just approved a matter of a few months ago by the FDA, approved for patients with advanced GIST who have failed on the first, second, and third line treatments, including imatinib. This drug acts in a very different way on the KIT protein. Instead of binding where ATP binds, the way that imatinib and the other drugs did, it binds to a quite different region of the protein, the so-called switch pocket. I won't go into that in detail, but the cartoons show you that the switch pocket is a region of the protein that's required for this change in structure, conformational change, that is the switching on of KIT into its active form. So it binds at a quite different region from the ATP binding pocket where imatinib binds. So as a metaphor, I can say that imatinib cuts the fuel line to the KIT engine. Ripretinib jams into the piston itself and shuts off the engine in a different way. So it's really excellent to have a drug that has a quite different mode of action in inhibiting KIT. One might hope that cross-resistance to the two drugs is a lot less likely. So Quinlock was developed in the last few years. This is just a timeline, which I won't go over in detail, but it shows you the rapid progress in implementing the registration trials and the approval of ripretinib. We're right in that timeline right now in 2020, and we hope that marketing will be and general use of ripretinib for imatinib-resistant drug just will become widely available in the next few months. A couple of other drugs I wanted to mention. Avapritinib. This was developed by a company called Blueprint in Cambridge, Massachusetts. It used to be called Blue 285. It's now marketed as AvaKit. This particular drug, we had hoped it would be generally useful in GIST. That does not seem to be the case, but it is very useful in a subclass of GIST, which I've referred to before, the so-called PDGFRA D842V mutant GIST, which is a subset of GIST, but quite a common subset. And Mike Heinrich, whom good friend of the LifeRaft group, one of the best experts on GIST in the world, he said it's tremendously rewarding to be able to offer for the first time a highly effective treatment option to patients with this particular mutant form of GIST. It will really become the new standard of care for that particular subset of GIST. Okay, so there's a little further detail about avapritinib. Won't go over it in detail, but again, just to emphasize that to use avapritinib effectively, we have to be able to identify the GIST patients who have this particular mutation. Failure to treat this subgroup, if there is inadequate mutational testing or gene profiling, that would really be a missed opportunity. So the availability of this drug is now a very strong additional motivation for universal mutational testing with GIST patients. The last drug I want to make reference to is a drug called larotrectinib or Vitrakvi. This has been introduced very recently. This is a drug that probably is only going to be useful for extremely few patients with GIST. It is a drug that is designed to inhibit the so-called NTRK mutation, NTRK. This turns out to be a very rare mutation in GISTs, but it is known. A few, maybe less than 1% of GISTs, but the occasional GIST is driven by this mutation. And in fact, a lot of non-GIST tumors are driven by NTRK. So although this will only be relevant to a very few GIST patients, it's turned out to be a highly effective drug for those particular unusual mutational classes. So I mention it for that reason. And there are other drugs on the horizon. So the final drug I'm going to mention is one that doesn't even have a proper name yet. It's developed by the AstraZeneca company. It's called AZD3229. It's only been discussed in the past couple of years, but it turns out to be a very potent KIT inhibitor, much more potent than imatinib. And it's hoped that this drug, the companies developing this drug, are hopeful that it will become the so-called best-in-class inhibitor. Ultimately, perhaps it may even have potential to replace imatinib as the first-line therapy for GIST. However, it's early days yet. For now, this compound is really just a chemical compound. It hasn't even been formulated into a drug yet, but we're looking forward to that happening over the next few years, and maybe this will turn out to be another highly effective drug for GIST. Okay, I've gone on a bit, but I've now gone through the material I wanted to cover in this GIST 101. If you do have questions, I'd be very happy to take them, and you can email them to me at this email address for Life Raft Group Canada. Thank you very much for your attention.